screen. Can you all see? I'm going to put it full screen.
Yes, that looks good.
Okay, perfect. So yeah, so I'll share, as Deborah mentioned, I'll share some information about EEG. In particular, I don't think I'll be talking for an hour. I can also talk a bit about the project, but I think the most important at this point for you, from what I gathered from Deborah, is really for people who want to use the EEG, getting some more information about it, so that then you can use it wisely and yeah, get the right information from it and understand a bit what it can do, what it can't do. So maybe I'll talk for about half an hour, maybe a bit more, and then you can ask questions. We can have a discussion if you like. I'll try to answer to them as much as I can. I'll talk a bit about the Muse system. I actually managed to get my hands around around the Muse, but I'm not an expert on the Muse system. But trying it out helped me figuring out a bit what can we get from it, what kind of information we can get from it, but also what are the potential limitations. But we can have an open discussion, particularly if some of you are already using it, we can discuss that. So I'll have a bit of a theoretical discussion, presentation first. I'll try to explain what EEG is, what it measures. Then I'll talk about what's the signal we're looking at, what are the alpha, beta, all that, what is it? And then I'll talk about artifacts, potential issues with EEG that you need to be aware of when you're using it. So a bit about DOPS first. So maybe you're familiar with DOPS, maybe you're not. But so as Deborah mentioned, we're part of the University of Virginia. We're part of the Department of Psychiatry and Neurobehavioral Sciences. It's kind of a mouthful, but that's basically the psychiatry department of the School of Medicine. And we're a research division, and we study extraordinary experiences, in particular experiences that kind of question the mind-brain relationship. So maybe it started with the study by Ian Stevenson of young children who have purported past life memories. So we call it court case of reincarnation type, near-death experiences, out-of-body experiences, mediumship, ESP, and psychokinesis. And we 100% privately funded. We don't get really money from the university or anything. So I mean, if you have money and you want to give it to us, you're more than welcome. So we function thanks to generous donors like Ray Westfall and the Williams. And so about myself, I'm directing the Ray Westfall Neuroimaging Lab. So we have a very nice neuroimaging facility where we can measure an array of physiological signals, including EEG and FNIRS, which are two different neuroimaging modalities. They're pretty cost efficient compared to a scanner, for instance. It's several, it's a very heavy piece of machinery. The MEG as well. So EEG next to it is like a little toy, even though those ones, they're like 128 channels, and so it costs several tens of thousands of dollars, but still it's cheap compared to if you want to use scanner, that's an order of magnitude, 2 orders of magnitude more expensive. First I'll talk a bit about what is an EEG, what do we measure. So this is going to be a bit theoretical, but I think it's important so that you understand what is this signal that we're measuring, right? So the EEG actually, it's kind of a simple system. It's a voltmeter. So it just measures volts between 2 points. And so 2 points on the scalp, right? So each electrode, basically, you don't measure brain activity here, right? You measure the difference of potential between this place and another place, which is the reference. right? And the potential, what it is it's what makes electrical charges move around, right? It's a bit like the equivalent, it's an analogy that people use a lot is a river. So imagine the electrical current is the flow of the river, and the difference in elevation is the potential. So it's kind of the potential energy that you can provide to electric charges, and because of this potential, then you have current happening, right? So you don't need the current to have a difference in potential in volts. But if you were to make a circuit, then the charges would move around, right? So that's what an EEG is. It's really plugging in a voltmeter and recording it across time, and that's what it is. But so why a voltmeter? Why does it give us any kind of information? So as you know, probably, the brain is composed of neurons, right? And the neurons they fire spikes, right? So that's how information is transmitted throughout the brain. It's spikes. The spikes are differences in potential, um, so differences uh, in charge, if you want. And they have so those spikes, they're very brief in time, right? So those are basically units of information. So this is recording from this second neuron, let's say. So you've got a presynaptic neuron and the postsynaptic neuron. Presynaptic neuron means, so you've got a synapse. It's a very ugly neuron. It's not very realistic, but you've got the soba, which is the sort of head of the neuron, and then it transmits information through the axon. And the end of the axon, you've got a sort of gap between neurons. And when the neuron fires, so you've got the spikes, the spikes travel through the axon. And then once it arrives here, it releases some molecules called neurotransmitters. And those molecules, they will land on receptors on the postsynaptic neuron, and they will what they will do on this next neuron is that they'll change the charge of this neuron. They'll change the balance of charges. So if you've got an excitatory signal, that will tend to make the second neuron fire afterwards. Then you'll have these little changes in potential. So the neuron has a certain potential, and then when the presynaptic neuron fires, when you've got spikes and neurotransmitters arriving here, and it wants to excite that neuron, it depolarizes that neuron, and when it reaches a threshold, poof, it fires. It sends a spike, right? And so what we think and what it looks like the EEG is actually measuring is those depolarizations here, right? So you don't really measure the actual spikes that the neurons generate, but you measure the depolarization or the polarization, because if you have an inhibitory signal, it would overpolarize, if you want, the uh, the the neuron, and it would change the charge in the opposite manner, so it won't fire, right? So this is just one neuron, but when you have many neurons at once, and and I'll talk about that, if they're organized in a certain way, then maybe you can measure it at a wider scale. So this is a cut of the brain. So it's a cut, it's called a coronal view. It's cut like that, right? And you can see the folds of the cortical surface. So basically, the cortex is like it's a surface. It's like a kind of a map, and it's flat. It's a surface, and then the neurons they are on this the like the soma of the neurons, if you want that. The gray matter is here. It's as you can see, it's folded. And let's say I take this part of the cortex, and it's composed of layers. We've got 6 layers, and you've got some layers that have some kinds of neurons that are a bit bigger, and they're called pyramidal neurons, that they align vertically. They're aligned perpendicularly to that surface, right? And so when a neuron, so this is this part of the neuron, this little dendrites, right? And then they get some input from here, and then this is the head of the neuron, and then they send information down there. And they can go outside. So this is the white matter, and those are tracks, if you want. The neurons sort of send information through there, and then maybe it will land in a different area of the brain. But so when a neuron gets some information from a presynaptic neuron, then it will basically, if it's a excitatory signal, it will depolarize, sorry, here. And then compared to here, the soma, the head of the neuron, it'll be plus. So it's relative, right? If you go to depolarization here, in comparison, this is going to be plus, right? And so the corticals, so those neurons, they're all aligned, right? And what's nice, and also they can be excited, so they can be excited at the same time. So you can get a bunch of information that arrives, and it excites all at once a bunch of neurons in the same area, right? So when you have this happening at a larger scale, so many neurons, you can thousands of neurons like that, all aligned in the same, or tens of thousands of neurons, or more than that, in the same area, depolarized at the same time, then it sums up the field that's generated. And that's called a dipole. When you have a negative charge next to a positive charge, it's called an electric dipole. And then this generates, let's say, a field that will be measurable further away in space, right? So what happens is that this little depolarization that we have, this little dipole that we have, is there, and then this is to model, let's say, to illustrate the skull and the scalp. Then basically you have a depolarization, you have a minus, you have a change of electric field. It generates a electric field that is measurable on the scalp. So on the back, here is going to be more negative, and then in the front it's going to be more positive. Yeah. So then if you put an EEG or voltmeter somewhere, so you've got your reference here, let's say, and then here above your dipole, so you do this signal, this potential minus this potential, you get something negative, right? So what the EEG does mostly is measure, it measures differences in potential between 2 points on the surface of the scalp. And mostly it will measure cortical activity. And even I can add a bit more to that. It will measure cortical activity mostly that is close to the skull, and that is perpendicular. Sorry, on those parts of the cortex that are parallel to the skull. So those dipoles that are perpendicular to the skull. It doesn't mean that you don't measure here. I mean, every dipole basically will contribute. So you imagine you have dipoles everywhere all on the surface. So this is just one dipole, right? But you always have brain activity occurring all the time at any time point. So the whole surface of the skull of the cortex is full of dipoles. And so you sum up all this activity, all these dipoles, and then you end up with a pattern of plus and minus charges, if you want, on the surface of the head. And you have to understand that the electric potential basically is has to go through different layers. So you've got the skull, you've got the skin, um, you've got dura, you've got a lot of layers, and so that will lose some spatial information because of that. So basically, that's what you measure with EEG, those dipoles. And then you've got parts of the brain, subcortical structures, where the neurons, they're not aligned in that way. It's kind of a mix of neurons oriented in many different ways. So those are not likely to contribute much to the signal. And you've got deeper structures, even on the cortical surface, here, for instance, or here. They're very far from the skull, and you won't measure much of it. So this is, in a nutshell, what the EEG measures. And this is what the EEG signal looks like. So it's a voltmeter. So here we're looking at a specific time point, right? So imagine we're just at one time point, and this is what our dipole at that point. But then what the EEG does is that we measure through time, right? So across time, and then often you have several channels. And again, each channel is not one channel. It's always a channel with a reference. It's always a difference between 2 points. And what people do usually is that if they have many channels, they might take as a reference the average of all the channels, right? But it's important to realize that it's not 1 point that you measure. And so you have each channel here, time in seconds here. So this is 1 second. You can see, you can observe some signals. So this is probably alpha because it's occipital, and it looks like you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You have about 10 oscillations in 1 second, and that's very likely to be an alpha signal, right? And then it's a potential. It's in microvolts, so it's very small. So just to let you know, so this is just from a paper, 2017. So you can have enough information in this signal. So let's say this, and I'll talk about artifacts, but it's a very noisy signal, and you can see it's kind of, this electrode, it looks like it's a bit noisy. It is actually, and the signals are tiny, and it doesn't look that there's much information. It's very noisy. It's very noisy. And so what people do usually when they do research with EEG is that they have to repeat many trials, or they have to measure during long periods of times, so that then they can get some useful information. And this is an example of image classification. So they showed people images like that, so of different categories. So body, face, human, non-human, natural, inanimate, natural, artificial. And then they used the machine learning algorithm on the EEG, and I don't know how many sensors they had, but probably quite a bit, at least 64. And then they trained a classifier so you show many images, like hundreds of images to people, to one person. Let's say you do it with one person, and then you take a bunch of the image, like say most of 80% of the images, you use them to train a classifier, and 20% you use that to test the classifier, and you plot the accuracy of your classifier across time. And you can see that using this kind of method, you get 65% accuracy in classifying the category of the image that the person is shown, but at 100 to 200 milliseconds, and then it after you show the image, right? So it's very short, right? So you show an image, a visual image, so it's something real, right? And then the classifier is pretty good. I don't know if they were binary. It looks like it was binary, so it's maybe inanimate versus animate, because it's 50% before, so it's like just a binary thing, and they get 65% accuracy, and that's probably on one person, and then they take many people, and then they average across people. But the classifier is learning for each person, most probably. I don't remember the details of that specific study, but it's just to give you an idea of, even for a binary classification in very controlled conditions and with two categories and probably quite many sensors, you don't get much better than 65% accuracy. So you basically 15% better than chance. So you 15 points higher than chance than um, when you use that method. So you can look at those are different methods. So MEGs is measuring the magnetic field instead of electric field. So this is a big machine. It's better at it because the magnetic signals that are generated by the neural activity, they are not blurred by the skull and the scalp and the um, uh, like the EEG is. So you get better signal basically. You get a better spatial resolution. But still with EEG it works. It works. All right, so we see all these squiggly lines, and so that's what's called the temporal domain representation of the electric potential of the EEG. But often, so if you use in particular the Muse system, and maybe you read some things in the literature, you talk about alpha, you talk about beta. So I'll talk a bit what is that. So um, so what I showed earlier is the temporal domain um, representation of the signal. You've got something called, I mean, if you've done signal processing or engineering, you know about that, or mathematics, something called the Fourier transform. You can decompose any signal or represent any signal, temporal signal, into a sum of frequencies. Um, what you do is that you will represent as a function of frequency the contribution of each sine wave. So basically, you take a signal, and actually it's quite easy to see if you look at this one. It kind of looks like a sine wave. It's not really a sine wave, but it kind of looks like a sine wave. And you can see that, well, sometimes it wiggles a bit up and down. So you can imagine, okay, maybe there's a slower sine wave, and this one is the one you see with the ticket line at 10 hertz that I counted earlier. They're riding maybe a slower wave on top of it, right? So you can see kind of maybe with this intuitive example that this signal could be a sum of different sine waves, right? But you can always decompose a signal in a sum of sine waves. It's a mathematical thing you can do. And the way you represent that, it's like, so say you have this sine wave. So this is a pure sine wave. And in 1 second, you have 2 cycles, right? So this is the time, 1 second, 2 cycles. So what you'll do in the frequency domain, you'll represent this. So you have the amplitude, which would be this height, right? So the height of your sine wave. So you choose the height, and then so you said it's 2 cycles per second. So 2 cycles per second, and you say it's only one sine wave, so you just one here. You just put a value here, and all the rest is 0. And then if you have a more complicated signal, which is the sum of two sine waves, so a slower wave and a faster wave, then you have, so it's a sum of two. You've got two sine wave, two contributions. So the first is a slower, so it's got a lower frequency, right? F1 is lower. Let's say it's got a larger amplitude. And then the second one is a bit faster, and it's got a small amplitude. So this signal, which is the sum of two sine waves, can be decomposed like this and represented like this in the frequency domain, right? And that's the spectrum. And you may have seen representations like that online in the literature, where you have, this is exactly that, basically. It's the power, which is the square of the amplitude, as a function of frequency. So you represent the contribution, or if you want, the height of those sine waves as a function of frequency. So instead of having discrete, here we've got only two. Here it's basically an infinity of sine wave of sine waves. Yeah. I mean, it's not an infinity because it's digitized, right? But in theory, it's an infinity. And you can represent the contribution of these sine waves across frequencies, right? And then researchers kind of arbitrarily define some domains in frequency, and those are the different frequency bands that we talk about and that you probably heard about. So you've got delta, which is lower, very low, below 4 hertz. Theta, which is typically between 4 and 7, 8 hertz. Then you've got alpha, which is between 8 and 12 or 13, or 7. It depends, but typically it's 8, 12. Beta is up to 30, and then you've got gamma above that. Right. And then you got high gamma up to 100 and above. One thing you can notice, it's not really noticeable that much. I don't know if maybe I have another slide after, but as you go, as you increase in frequency, the signal becomes smaller and smaller. So the higher the frequency, in particular when you go higher than 20 or so, the smaller and smaller the amplitude of the signals of the sine waves are, so the noisier they are. So the more difficult they are to measure, and the more susceptible to noise they are. But the most prominent signal by far is alpha in general. And also theta and delta, they tend to be quite high. But alpha is very prominent, in particular occipitally and occipital locations. It's very prominent. It's very easy to measure. And so people try to look at, so because you're interested in outer states probably, so they calculate the spectrum for different conditions. And here, for instance, so they looked at meditation, and they found that the spectrum actually is different for those meditators compared to controls. I don't know the details of that, but you can look at that publication if you want. And here you can see it more actually, that so this is it's a log scale, but you can see that the power decreases as a function of frequency. Okay, so that's the spectrum. So now we understand what delta, theta, alpha, beta, gamma are. Something that you need to realize is that the brain signals, they're very, very small. So we're talking about microvolts, and we're talking about the sum of the activity of neurons, and it's very, very small. And next to that, you have other sources of changes in potential that's maybe way bigger, and that might hide your signal, your brain signal. And you've got different sources of these artifacts. Some of them are physiological, so they come from the actual person that from which the measurement is done, and some are non-physiological, and they can come from either the recording device or maybe from the outside. Right. So the most common artifacts, and they'll be there in most of the recordings, will be ocular artifacts, so eye blinks, eye movements that will definitely show on the signal. Muscle artifacts as well, so facial contractions, skull contractions, the tongue. So if you move your tongue, you'll see signals as well. The tongue is a muscle, so muscles also work. They also work through electrical activity, right? So they generate a strong signal when they contract. If you talk, if you move, if you swallow, that will show on the EEG. The heartbeat can be measured as well. It's not always there, but in some people, some recordings, you can actually see it. Respiration can show as well, and sweating as well will show, because sweating will will basically can kind of short circuit. It will change. It will change the. It will help electric charges maybe from moving from one place to the other without having to go through the. And then you've got non-physiological sources. So typically, maybe not with the Muse, which is using dry electrodes, but many EEG systems, more professional systems, they use gel or saline solution to make contact with skin, and the properties of the gel will change as a function of time. It will dry, it will move a bit, so it may modify the contact with the skin, and therefore it will change the way the signal looks. If you pull a cable, so you may move the electrode a bit, um, and you can have a bad connection as well with an electrode that the connection varies. And then you've got electromagnetic coupling, right? So if you've got a circuit and you've got you're measuring so to measure volts, you need to make charges move around, right? And you need to create a circuit. And then if you've got something that creates a magnetic field around, and it will make those charges in your circuit move, and it will be recorded as a change as a current, and then as a change of potential. So power noise will be definitely measured in your circuit, even if you. So typically you measure there's a battery systems, but still you have very strong. Every house has a uh, power cords everywhere, and so you can see at 60 hertz these sine waves, and they will probably show in your signals. So you need to be aware of that. If you got a cell phone next to the system, it will probably generate some noise as well. So we need to be conscious of all that, and that's why, for instance, that's partly why we have a Faraday cage in the lab, is to avoid these electromagnetic coupling noises. Right. So some system there will be, I don't know about the Muse, and the Muse fortunately doesn't have cables, so it may reduce electromagnetic coupling. Um, but um, but yeah, if it's something that will happen. So I'll go through a few artifacts so you can just get an idea of what it looks like. Um, so those are found online. They're very common things that you will see when you look at an EEG. So you can see here, so FP2, F8, so those are frontal electrodes, right? Probably those are bipolar, so they're looking at FP2 minus F8, and it doesn't really matter. But so you can see that this is the brain activity, it looks very small, and then you've got those big jumps here, right? And those are eye blinks, right? And you can see how much bigger they are compared to the EEG signal, right? So when I run experiments, I look for, so there are methods to remove eye blinks with ICA. It's pretty efficient. Um, but you need to take this into account because they will hide your signal, and they also tend to be in the same direction, so they will distort your signal. So sometimes you have noise that sort of cancels each other, cancel itself, because sometimes it's in one direction, the other one in the other direction. And so when you average the signal over many trials, when you do ERPs, which are a way to average the signal together, sometimes it can work, it can average itself out. But the eye blinks, they always tend to be in the same direction, so that's annoying. Here, the participant control the facial muscles, and you can see those very high amplitude spikes, right? It's very, very strong again. So you've got the activity looks very small, and then you've got this very strong signal. So this is very easy to see, and you would remove that part. You could also use a low pass filter that would remove high frequency parts of the signal. So you need to be very careful when you look at beta or gamma, because this is the window in which the signal will, sorry, so those frequencies, beta and gamma, will be contaminated by muscle artifacts, high frequencies. Whereas eye movements, eye blinks, it will be more in the theta range. Here it's an example of so a participant is talking, so you can see like muscle movements, like facial muscle, and probably from the vocal cords as well, and then he's moving the tongue. They're moving the tongue, so you can see those low frequencies, say la 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 la. You can see how it's generating those. And again, this is the EEG signal, tiny wiggles, right? And then you've got the artifacts that are way bigger, and they're hiding your signal altogether. Here's an example of electrodes that are not very well connected, or that they are noisy for some reason. This is probably a bit noisy as well. You can see it's like those little spikes here. This is quite common. Those square shapes. So again, we're in frontal electrodes here. Those are eye movements. So the person is probably moving their eyes left and right here, and you can see on those. I don't know if because here F7 T3. Yeah, it's not a maybe best example. But so typically you'll see an inversion. So when you have the electrodes left and right, on the left and the right, if the person is moving their eyes left and right, it'll look like it will be symmetric, and and you have those square shaped signals as well. So this is just an eye saccade. So the person is moving their eye, and so you've got to jump because the eye is also polarized. So it's like a big dipole itself. And when you turn your eye, when you look right, your whole eyeball is turning, and so basically the dipole itself is turning. And so that's measured, and again, it's orders of magnitudes larger than your actual signal, than your actual EEG signal. Okay, so this is a little tour. This was a little tour of EEG artifacts, and I'll talk a bit about the Muse. So I'm not, as I said, I'm not an expert on Muse at all. I played a bit, a few minutes with it this weekend actually. It seems to be it's really cool system actually. I think there's a lot that can be done with it. It's very straightforward. Just put it on your head, and it's great. So from what I understood, you have only a few electrodes on the front of your head, right? So you measure those frontal sites. From what I understood, but I don't know if that's correct, the reference seems to be in the frame. So I'm not too sure. From what I read, but I don't know if that's correct, it seems to be more on the ears, the reference, which is fine. And there's an accelerometer, there's gyroscope sensors. So I think to some extent it will probably try to remove some of the noise I talked about. But I'll show you, I played with the Muse 1. I don't know about the Muse 2, but with the Muse 1, I played a bit and I tried to see how robust it was with noise. But so the app, so there's an app basically that's associated with the system, and it gives you exercises, meditation, and it gives you a score of relaxation, and it tells you if you're very relaxed or not. And everything is proprietary, so you don't really know how it does things under the hood, which is a bit annoying because you don't know what it does. But there's a way, if you're tech savvy and you want to go into details, there's a way to get the raw data and to process the raw data by yourself. So you need to subscribe. There's an SDK, so it's a way to software development kit. There's a way to through software to get the information, the raw data from the Muse system, and then you can process it by yourself and do your own experiments and all that. Because the information that you get is not very detailed. So I played a bit myself. So I was pretty relaxed. I was at 60 BPM. Well, I was at rest, let's say. So this was my rest. So I did not move. I just waited for a few seconds. So that's what the signal looked like. And then what I did is that I did this. I moved my eyebrows up and down. So it probably moved the connection of the electrodes. So you can see all the green. So those are different sensors. So there are 5 sensors, and green means the good connection, right? That's why I moved my eyebrows up and down like this. I just did this, and you can see how it moved the signal. And those are artifacts, right? Compared to this. Here I did just eye blinks. I just blinked many times. I did this like this without moving my eyebrows, and I just did this. And you can see again, you've got very large signal. Here I move my eyes up and down, and here I did a single eye blink, and you can see how it shows. Here I smiled. I did not move in the upper part of my face, but I just smiled. And here I contracted my upper body muscle. So I just did this like this. I sort of contracted all these muscles here, and you can see these very high frequency signals. And here is one of my hidden talents. I can move a bit my ears, and that's what happened. So from what I read, but maybe that's the Muse one. From what I read, there's some some artifact reduction, but still, so in the Muse one at least, it seems that it still lets a lot of noise pass. So one thing, if you want to use the Muse system, you need to stay still, and you need to not move your facial muscles. You need to not speak. I didn't try speaking actually. I should have done that. But because if I move my eyebrows, if I smile, and I contract my muscles, my upper body muscles, it makes that noise. Probably when you speak, it also makes those noises. So you want to stay still when you use the Muse. So this is the time domain. So this is time, this is amplitude, and I don't know how it's calculated. It looks like maybe it's the average signal between all these electrodes. I'm not sure. It doesn't say, or maybe it says somewhere, but I didn't see the information. And you can also have the spectrum, a sort of representation of the evolution of the spectrum in time by frequency band. So each color is a different frequency band. So green is delta, which is low frequency. You got theta in light blue here. So this is theta. Alpha is this blue here. Beta is this purple, dark purple, and gamma is the light purple. And so at rest, it's pretty low here. And then I did the same thing. I moved my eyebrows up and down, and boop, it peaks. All the frequencies peak, and because well, it moves up and down, I guess, and the low frequency, and then you can see these high frequencies. So yeah, those are eye blinks. So eye blinks, they're quite slow, right? So you can see that the low frequencies, delta, theta, and alpha go up. But the other ones, they don't go up so much because it's high frequency. And that makes sense. Same for the eye movements, up and down. So I just looked up, down, up, down. And again, the low frequencies peak, but not the high frequencies. So that makes sense as well. Smiling, for some reason, slow frequency. Maybe I didn't smile long enough. So I just did a smile and then stopped. So maybe that's why you have the low frequencies only that peak. And then I did that upper body contraction. So it's not so bad actually, but you can see that the power increases in all frequencies, right? So if you rely on this representation, you can see that also it is susceptible to noise. Because I was thinking maybe these representations are maybe more raw data, and those are more processed. But it seems that it still lets quite a lot of noise go through. So I don't have more to show, but yeah, the bottom line is, so I think those systems, they're great because they're so cost effective. They're really I'll just show this picture. Um, they're um, they're not expensive, or they're quite affordable. And and in particular through the SDK you can get to the raw data, so that's great. And so it's quite flexible in many ways. But you still need to be wary of, you need to be careful of what you're doing, and you need to basically know what you're doing and be aware of these potential pitfalls, because you don't want to interpret things that are artifacts, because the artifacts are so prominent. As I showed, they are actually usually larger than your actual brain activity. If you rely only on those very slow, um, on these representations here, you can't really know what you're measuring, right? You can only assume if you've got a big change like that, it's probably that's going to be an artifact. Um, but it's hard to interpret. So this representation is good because you can understand a bit better. But if you can get to something a bit more raw like this, where you can see each sensor and not the average across sensors, that would be better. So if you're using the Muse, I would encourage you to try and subscribe to the developer toolkits, so you can go into the details of the signal and understand what's going on. And then again, you need many trials, you need very clear hypotheses, and we can discuss that after. So then you can look at the parts of the signal you're interested in, and contrast different conditions to be able to to make conclusions and inferences. And that's it. So I don't know what time it is. It's so how long did I talk for? I took for almost an hour. Okay, I wasn't
It's,
expecting me for. I
no,
was expecting we took for an hour.
that's great. I'm sure we have a lot of questions. Thank you so much. That was very, very informative. And well, I'll just start calling on people that have questions. Um, and Andrew, you're on mute
You hear
there.
me?
Yes.
I had a few questions. I'm not sure all of them are relevant. First, thank you for your presentation. I found it very interesting. The readings that you were taking, were those done during actual remote viewing sessions, or was it just you were just measuring the brainwave activity when the person's trying to be still versus physical movement? Were these actual remote viewing sessions where you were measuring this, or is it just brainwave activity compared to physical movement? And then I
Yeah,
had,
it was just for myself doing that. I was just resting and then I was moving. I was not doing remote viewing. I cannot do remote viewing myself.
and then it was interesting because you had a picture of the brain, and you were kind of showing where synaptic connections occur. And like a phenomenon of remote viewing, especially for a beginner, is like sort of beginner's luck, where you make that connection kind of early within the first few sessions, and then it seems to kind of taper off until you redevelop it. So I guess that's not that relevant if you weren't checking it during the remote viewing sessions, but because you had that picture of the brain, I was curious to see like what part of the synaptic connection within the brain was happening, especially at the beginning when you have that beginner's luck connection part of it. Like in other words, what part of the brain in that diagram that you had is most relevant to the accurate remote viewing sessions. But since it's just basically a physical, it's you sitting still and then versus movement, I guess that's not.
Yeah, I don't know. Like, I think no one knows what's the and actually we're starting a research project with Marina that Deborah mentioned, which is about trying to understand through the EEG, trying to characterize the neural signal, the EEG signal that is associated with remote viewing activity and associated with accurate impressions versus inaccurate impressions.
I
So my answer is I don't know. And the research that has been done on remote viewing and ESP more generally is all over the place. There's some hints that alpha may be implicated, but some people found an increase in alpha, some people found a decrease in alpha. So it's still an open question.
I found it really interesting at the end of your presentation too, that when you showed the different readings, like delta versus alpha, how when there was movement, the delta was the one that went to the highest, when that's actually the lowest brainwave speed. So it's measuring basically the spike in it. And in some practices in remote viewing, you pull back from the target to kind of reestablish yourself and then move towards the target again. I was kind of hoping to see this to help me determine like if alpha was more effective than delta, but that was interesting that the movement created a greater spike in delta, but that's only because delta is more still. When you're in delta, you're physically way more still. So any movement there would obviously show a spike on those charts because it's a lower frequency versus the alpha frequency is a little bit higher. So the movement's not going to impact it as much. Again, I'm trying to compare this to actual readings through remote viewing sessions. So it's a little bit weird, but
Yeah, no, I see what you mean. So when you move, so if you contract muscles, you'll typically have spikes in. So it doesn't show on the Muse for some reason, but although I think for some of
I don't
the
know that that was the Muse part of it, though, or maybe it was.
so typically if you contract muscles, you'll find it in beta gamma. You'll observe it in beta gamma. But sweating will be delta. You have very slow drifts or even infrared delta, very slow drift of your EEG. Eye blinks will be more on the theta, alpha. But yeah, if you move a lot and your EEG signal is going all over the place, it will also affect a lot the delta, the theta and alpha because your baseline signal will change slowly, but on top of it, you may have, say, muscle activity, and that's high frequency. So basically, all the frequency band may increase in power. So that's why it's very difficult to interpret. But yeah, I guess if you stay still, and things go down, then I mean, the power will go down because it won't be contaminated with all this noise. So you won't compare things that are comparable. So you want to compare your, say, while you're doing your remote viewing, if you still, you want to compare your remote viewing when you still with when you're not doing remote viewing and when you still as well, right? Because if you've got one condition where you move and the other one where you don't, you've got a confound, and the confound is, well, you're moving. So yeah, of course, your signal will look very different.
Yeah, and I did thank you for that presentation because that was interesting and helped me to see that the lowest one where your physical body is the most still. You're almost asleep. It's almost like you're sleeping in delta, and if the tiniest bit of movement, the delta is always the highest spike.
Mm-hmm.
That
So when
makes
you say,
sense.
yeah, I hear a lot. So when you're in delta or you're in alpha, so to me it's a bit of a, I don't know how you say in English, in French would say un abus de langage, an abuse of language, let's say. So you always have, and I can share again, you always have these uh, spectrum, right? And when you say there's more alpha, it's just maybe your peak will be a bit higher. And when you have a bit more, when you're in delta, maybe you have a tiny bit more delta, right? But globally, this won't change a lot. Those are between people, by the way. So between people, you'll have more variations. But within a person, when you're doing some activity, may have a little peak somewhere, but you won't change that much, right? You may have a bit more alpha, a bit less, but the shape overall will be the same. So, so, oh, I was not sharing my screen, was I?
No.
No, I was not. No, so what maybe I will share it again. And just so you see.
And everyone, don't worry, we'll get to everyone's questions. We have plenty of time, so we'll make sure everyone has their questions
Yeah, sorry, I'm a bit long, but I think
Oh, that's okay.
Yeah, so here you can see the spectrum. So the spectrum won't change much between different activities you're doing. It will change a bit, but so here the spectrum is a bit different, but it's also because is measured between different people, right? But to me, saying being in alpha or being in delta is it may subjectively it may feel very different, but it doesn't mean that you have way more delta or way more alpha than before, right? It may be a bit of a change of um, of power, and I would personally not use those terms because they're probably a bit simplistic in the sense that um, I'm not. I mean, I don't know where it comes from, but I do hear it from time to time from people. Um, and I think it's making a bit too much of an equivalence between having more of one frequency and having one being in one subjective state, right? All the frequencies are always there, and it's their relative contribution that will change a bit, right? Um, and making the link between the subjective state and the change of activity is really hard. And for instance, I looked at this a bit of meditation. I'm not an expert in that, but so for instance, they're doing meditation, and so some papers found that posterior alpha increases, but also for very experienced meditators, apparently decreases. So it's complicated. What I mean is that it's complicated. So using terms such as being in alpha, being in theta, it may be overly simplistic. That's just what I'm saying.
It looked like the chart was just showing that it's not beneficial or not beneficial, but the difference in physical movement when you're in the delta base state, as low as you can go, and then there's movement, the spike is just the difference between the two, only because you're coming from a lower place. So the brainwave speed is already at the as low as it can go, and then a movement's going to impact that more than it would if you were in alpha, where you're not as low as you can go. So it's going to spike up, but not as much because you're already not as low. That's all. I'm trying to learn, the chart.
Yeah, yeah.
You didn't do this during a remote view session, but I just want to say thank you for your research, and I definitely got quite a bit out of it. I appreciate what you showed us. Thank you.
Sure, thanks.
Some of these, they're just getting started with the remote viewing research. So in a year or two, they'll be able to come back and really tell us a lot more of what they found when people are remote viewing and what their states look like. But let's hear from Diqing, and then we'll hear from Dale, and then from others.
Okay, thank you, David. Thank you so much. And I learned quite a bit. So I have two simple questions requiring only a numerical answer. And a third one might need a little bit more elaboration. The first one is that your 128 channel EEG, what's the highest frequency that you can process? And from your spectral, you showed it looks like 125 Hertz. Is that the highest that you can process?
Yeah, I think personally I wouldn't even go that high. I typically don't. I would typically low pass filter below 40. Like I would not go higher than 40 hertz or 50 hertz. Maybe 60 hertz in the US because you got the line noise at 60 hertz. In Europe, I wouldn't go above 40 hertz because you got the line noise at 50 hertz. Because as I showed, the spectrum is the amplitude is really low as you go higher. So you're gonna measure mostly noise. So maybe if you got loads of data and you know that you have something you really know that it's occurring gamma, you can collect a lot of data, and then maybe you can go up to 100 hertz, and that would be fine. So I'm not saying you cannot measure it. I'd just be very scared of of interpreting anything that's above 50 hertz
Okay,
because it's
thank
so
you.
noisy, and also it's in the muscle noise range, right?
Yeah. The second one is also related to that. So what is the smallest frequency difference that your 128 channel EEG can separate For example, can it separate the 0.01 hertz frequency differences between the spectrum, or was that too out of the range? So I'm interested in knowing the spectral resolution of your EEG processing.
So the spectral resolution will depend on your sampling frequency, right? And the biosemi system that we have, depending on how many channels you have, you can increase. So if you have less channels, you can increase the sampling frequency, I think. Typically, I think you can record up to 1 kilohertz. Yeah, 1 kilohertz, and I think it might be able to go higher than that. So yeah, I don't know exactly short answer is that I don't know, but you can also increase your resolution by padding, I think. So you can add zeros to your signal, and that increases your frequency resolution. But yeah, that will depend on your sampling frequency mostly, and you have tricks to try and increase it, maybe by removing a bunch of electrodes so that you can increase it.
Okay, thank you. The third one is that I saw that you have 8 by 8 the FNIRS. I don't know which company produced that. I played with the NIRS X FNIRS a little sometime when I was doing a sabbatical, and I'm interested in. So I don't know if you have any data from that, but I guess whatever that measures the sampling depths or the range of the cortex might be different from what EEG can measure. So I don't know if you have any data, but what's your projection? So how would the information from FNIRS and the EEG, if they would correlate with each other or not, or is there any insights on that part? Because that's really interesting.
Yeah, it's a good question. So we have an FNIRS system. To be perfectly candid, I do not have much expertise in FNIRS yet. So the company so we have a NR Scout system from NRX, and I think it's kind of an older system. So I don't think they produce this particular system anymore. So for people who don't know, the FNIRS system is very different from the EEG. It measures brain activity so it doesn't measure neural activity itself. It measures the oxygenation and deoxygenation of the blood, if you want, in the head using infrared light. So it shines infrared light, and then it looks at the refracted light and changes in the color, if you want, of the light. That will be an indication of whether there's oxygen or no oxygen in the blood. So you're not really recording brain activity, you're recording how the blood around the brain is, or in the brain is oxygenated. So the temporal resolution will be much lower because the oxygenation of the blood will change at a slower pace than the EEG. So you don't need such a high sampling rate. I think there's about 10 data points per second, something like that, or every 20 milliseconds, something like this. And it's a good question about the correlation. So the good thing about the FNIRS system is that it's much less sensitive to noise. So the muscle activity in particular will not affect your signal as much. If you pull an electrode, yes, it will affect because you'll break the connection with the skin, but noise, like muscle noise in particular, will not affect it. The eye movements will not affect it. So it's much more robust to noise, and that's why FNIRS typically is used for developmental studies with children, because they move around a lot, they talk and all that. So that's why we have FNIRS in the lab. It's to be able to have the participants doing a task that may involve movements, and will be able, hopefully, to measure signals that are more robust to noise. And it's a less used method, so there's work to do in terms of interpretation of the data. But again, I'm not an expert, so I will not digress too much on that topic.
Thank you.
And then let's hear from Dale next, and then Dirk after that, and then we'll go to our chat after that, or anyone else can raise their hands. Dale?
Yes, right. I'm quite interested in the MEG devices, but also realize that that might be subject to geomagnetic field variations as well. So different kinds of activity in the local magnetic flux in the environment might be another artifact that affects the output of the device. I'm also aware at one time there was a facility at the National Institute of Health where they used MEG devices to evaluate epileptic seizures and other medical anomalies, but they used cryogenic devices for measuring the magnetic fields from the electrodes that they had. And they were using different places on the scalp and determining positions within the brain itself using that methodology. But that's some years ago, and I'm not following that lately. So I don't know if they still continue that or if you know anything more about that kind of research.
So are you talking about MEG, right?
Yeah.
So the MEG measures, so you can see the MEG as so the EEG measures electrical activity. The MEG measures magnetic activity around the head. And it's a much more sensitive system. So usually when you measure the MEG, call it the MEG, it's done in a chamber, a bit like the one we have, but maybe even more it's a Faraday cage as well. And yeah, it's very, very sensitive. And I remember, so when I was in Italy, I was doing a postdoc and I was working with MEG. And you had if a car, so outside, so it's so it's in a chamber that's electromagnetically shielded. If a car was passing by slowly, you could see the car, right? You could see it on the signal. So you have to filter, and there are methods to filter the signal that are not just filtering through frequencies, but you model the sources of the signal outside because you measure around the head, so you can sort of infer where the signal is coming from by a kind of triangulation, if you want. So the first thing you do with MEG is that you sort of model where the sources of the signals are by decomposing the signal in a certain way, and you try to remove everything that's coming from outside the head. And that's a critical component of MEG signal processing that you don't need to do with the EEG, because the EEG is a bit less susceptible to noise. But the MEG, as I mentioned earlier, the advantage of the MEG is that it's not, the signal is not smeared by the head and the skin and all that. So then you can when you try to infer the source of your signal, it's more accurate than the signal. So you can say with more accuracy, okay, the signal that I'm measuring, the sources in the brain are here. Versus EEG, it's a bit more blurry, if you want. But those MEG systems, they're very expensive. There are not very many in the world. I don't know how many there are in the US, but maybe not more than 20 or something like that, maybe a bit more. But there's a good, now there are new methods called OPMs, optically pumped magnetometers, and those will be cheaper. There'll be way cheaper devices, and hopefully they'll be more accessible soon.
Okay.
Okay, thank you.
And then Dirk.
Hi. So with the Muse, well, I don't necessarily want to make an assumption, but I'm wondering if you're good at meditating, would that also indicate that you would be, it's the same markers you would have if you were doing remote viewing?
I don't know. Probably. I mean, it looks like I know very little yet about the neural markers of remote viewing, but from my what I understood, remote viewers tend to get to a calm place before they actually remote view. So at least there'll be at, I guess, markers that are typical to rest, to being at rest and being calm. Um, but at the same time, I mean, there are many kinds of meditations as well. So depending on what kind you're talking about, that might differ. But I don't want to make any claims at the moment because I don't know. My short answer is
Okay.
I don't want to say something too stupid. So
So, but in general, whether say meditating or remote viewing, have you found the ideal when people claim, yes, I've been in meditation or I've been remote viewing, that the baseline, what is the baseline with, I don't have them all memorized, the alpha, the beta, the different
yeah,
things
so
that
when
they're
they
measuring.
meditate, as I mentioned earlier, so it looks like alpha increases, it looks like beta tends to increase as well. Um,
Oh, they increase.
yeah, no, alpha can decrease, but also increase in what did I write? It depends if they are experienced meditators or not. So it increases usually, but it can decrease as well if they're experienced. Um, so if you close your eyes, your alpha will increase automatically, almost mechanically.
Okay.
But so again, with experienced meditators, it might decrease. Frontal theta seems to increase when people meditate, and gamma seems to increase as well. I've got a widespread gamma increase. So yeah, quite a few changes that seem to be occurring. But again, depending probably on the type of meditation you're doing, you might find differences. Um, but yeah, again, I'm not an expert on that particular topic. But hopefully, when we're done with this project that I was mentioning, we'll try and
Process.
compare. Yeah, we'll try and compare what we see when people do remote viewing with. So we'll have a baseline condition. We'll have while people do the remote viewing, and then we can compare. So we won't have a meditation. Um, we will not have a meditation condition, but we can hopefully compare with the literature and see if we have comparable
I
changes.
I think I could see where if someone was remote viewing and they are getting the target, then you can compare that there's examples of receiving an accurate target with what sort of readings are you getting, and
Yeah,
then see if other people doing the same thing would get the same reading or not, or if there's a variation, a range.
yeah. My hunch is that um, we'll see quite a bit of variability between people and also sessions because because I've started doing some interviews. The information can be very varied. So the subjective experience of the remote viewing varies a lot. So sometimes it's very visual, sometimes a bit more somatic, sometimes it's some really abstract impressions as well. So depending on that, we'll probably get quite different signatures that will depend on the modality of the impression itself. Um, so that will be interesting to see as well, to look at. Yeah.
Lots of studying to do.
Yes, yes. So this is just a first
Okay.
step. It's not quite a pilot experiment, but it's going to be a sort of, I think, one of the first studies in this kind. So we're sort of walking a bit in the dark. So I think that will be a first step, so that then we can have maybe more precise research questions afterwards that people can tackle and we can tackle. But yeah, it's there's so much going on during a reviewing session. We're trying to simplify what we're looking at, but it might be a bit too simplistic. We'll see. We'll see what we can get from it.
Well, thank you. It's fascinating.
Thank you.
Thanks.
Any more questions?
I have a question. I think,
Thanks.
can I go? Yeah. So I have a Muse. I'm wearing it right now, the Muse 1. And I didn't immediately find it easy to use. And I stopped using it. And I only used it briefly, but I stopped using it um, because I was worried about privacy. And I know you said you're not an expert on Muse, but it has an app. It goes to the phone, which is right here. And I mean, I know you're not an expert on Muse. You told us you're not, but is Muse just recording us and are we giving them free data that they can use to do whatever they want to do with whatever they want to do with it? So that's my question. Do we have any level of privacy when we're using Muse, or are we giving them free data from us, the customer?
I don't know. The question request is I don't know, but you can probably find out this information in the terms of usage or something like that. It probably tells you whether they take the data or not and use it for some reason, for some other applications. So yeah, I would go back to the terms of use, and then if you can't find this information, I would contact them to know what they do with this data and whether they store it or not. They may not store it, but probably they do.
Probably they do.
Probably
That's
they
the
do, so
thing.
that then they
Yeah.
can run whatever algorithm on it. I mean, probably they don't. Most likely than not, they're not using the information. Let's say the personal information. They're probably, but who knows? Running it anonymously, and then they use whatever they test the algorithms on this data, stuff like that. But yeah, I would look at the. Are you worried about your about giving your data?
I am growing increasingly concerned about my data and who I am and how much information about me is out there. And yeah, I think it's a psychic thing. I think well, anyway, we're recording this, so I won't say too much, but it's my personal concern. Yeah. Yes, 100%. I'm concerned about my data, and I think about it a lot. And I'm still on Facebook, and that's it, social media
Yeah.
wise.
So
Yeah.
software development kits, I don't know how that works, but it's possible that if you design an app that uses the Muse system, you can get the raw data and then I don't know if the software development kit would plug into the existing Muse app or it would be able to just get the data directly from the Muse system. If it's the latter, it would mean that you're in full control of where the data goes. If it's the former and it goes first through the Muse app, maybe you're not in control of that.
So
But
you're saying I should create software
maybe,
to
yeah, you need to look into that. But that's what I'm
yeah, I'm not
saying.
gonna
That's what I
do
was
that.
saying during the presentation. If you
Yeah.
are, I'd say, probably if you're serious about trying to do research with the Muse, you probably need to develop your own software, or maybe there are alternative softwares that are based on the Muse that would allow you to do some more in-depth analysis or process the data offline because the data that the Muse is displaying to people, and I'm not sure you can actually download it that easily. Maybe you can, but it doesn't seem to give you much to work with.
Okay,
Okay.
we had a question from Lori, and then we'll go to Andy. Lori wrote in the chat, how does Muse compare with HemiSync?
I don't know. I haven't. I don't know what the HEMIC is. So my short answer is I don't know.
Okay. And then Andy, would you like to ask a question?
Sure, thank you. Yeah, my question was I put in the chat earlier, but I saw on your slide you had done research, or it looked like you guys had done research related to mediumship and other possibly psi-related things. Were there specific correlations in the brain state data that you guys noticed when doing that research? Because I feel like mediumship, receiving of information could be very similar to RV perception of data as well. So just curious if you noticed any correlations, because I think many of us are interested in seeing, oh, we could look for the certain correlation of data in our sessions, right? And maybe an ERV session or something like that. So
Yeah, so we have data that dates from before I arrived. This data hasn't been analyzed at this moment. For mediumship, so we have some mediumship data. For mediumship, there's a paper by Arnaud Delorme in, I think, 2013, and they found increasing gamma during
gamma,
the.
okay.
They also bit, so they do mention that they say, well, gamma, okay, we find an increase in gamma, but we know that gamma is potentially confounded by movement artifacts. So we're not sure if actually it's an actual gamma increase that we're measuring or if it's movement. So it's tough. A lot of even beta. So I was talking about beta. So sorry, I'm digressing a bit, but just to tell you how this issue of artifacts is rampant all the time in EEG research. So there's Yuval Greenberg, who's a researcher. It's more in MEG, but it applies to EEG as well. They were looking at, I can't remember what exactly they were looking at. I think it was a visual task, but they found some beta, and in the end they demonstrated that the beta increases that that was fine they were measuring was actually microsaccades, so movements, tiny movements of the eyes. So the eye is always, you think you're looking straight, but actually your eyes are moving a tiny bit, and actually that creates a signal that can be measured through the EEG, and people, researchers, thought they were actually measuring brain signals, but it looks like a lot of it may just be eye movements. And there is some indication that maybe in alpha as well, there may be some eye movements involved in that. And so it's difficult to really interpret and understand what to interpret those signals that we're measuring. And it's very, you need to be very careful, and people are making mistakes, even very experienced researchers.
Thanks. Yeah, I guess my only other comment is after hearing that, it's very helpful to know how important the artifacts are, and makes me, in my own personal maybe research with the Muse device or something, is doing more of an extended remote viewing where I'm very still, being very cognizant of like my eye movement, and then kind of seeing what might correlate or come out of those sessions. And also being, even with my eyes closed, I might do eye movements up to try to initiate an alpha state. So be mindful of that too, and keep an
Yeah.
eye out for that. So,
So
but
an eye
thank you.
movement,
Yeah, it's very
yeah,
helpful.
yeah. So looking at the time course, you will see your eye movement up. So you'll see how it's a bit more tricky with the Muse system because you only have so few electrodes. But if you have a full scalp, you can also look at the map of the change, right? And you can see that if something is mostly the very frontal electrodes that are affected, and you've got this kind of gradient across your scalp, so the frontal ones are very affected, and the further you go away, the less affected they are. Very likely it's an eye movement that is generating an eye blink. Or,
Thank, thank you. Yeah.
but yeah, you're right. The extended remote viewing might be more suitable for that. In particular, if you're interested in the remote viewing state itself, versus trying to look at different parts of your impressions, because then you need temporal markers to know when things happen.
John?
Yeah, switching gears for a second. I'm just a layman person reading cognitive neuroscience for 20 years, but I've noticed that some remote viewers are starting to use AI in attempting to solve the patterns. And I'm wondering what the status is of AI in your work and the use of solving these incredibly complex patterns, right, which the brain produces.
Yes, so people are using AI in different ways to try and understand or interpret what's going on in the brain. I've used it myself. So I showed actually some classification methods, if I showed this. So this slide, this is kind of AI based. It's very simple. I think they're using LDA. sort of simple classifier. It's not so AI, but it's not like deep learning. But so I've used it to interpret sentence comprehension, but using fMRI. People can now reconstruct, so they measure brain activity and they can now reconstruct what people think of, or what people read, or what people are visualizing or are seeing. The limitation in that is that you need to train your system ahead with the same person, because the patterns across people will vary, right? So it's very limited, right? You basically do an experiment where you show different pictures, you make the person read or watch a movie for an hour, and then you train the system on, I don't know, 45 minutes or 55 minutes, and then you test on the 5 remaining minutes, and then you can shuffle that. But you could not probably, I mean, maybe you can do it with another session, maybe, but you could not do it across people, probably. I mean, maybe you can now, I don't know, but you have limitations. But yeah, people use that. And people use AI as a model as well to try and understand how similarly the brain functions as an AI model. And to some extent it does, but in many ways it doesn't.
Do you think that AI is going to be required to really understand how memory and all these different functions in the brain take place?
I think it will help understand patterns, because the patterns are very complex, and what AI is good at is to find patterns that are associated with a meaning. So you can train a system that's your input, that's the brain signal, that's the variable we're trying to link it to. So then you can have a system that's trained to try and link those different things. So for semantic meaning, for instance, people have managed to read the brain and link the patterns in the brain to actual words, let's say. But the problem with these systems is that it shows them that there are specific patterns associated with certain meaning, but it doesn't make us really understand what those patterns are. So there's still a layer of interpretation that needs to be added to that. So it can help us, but it won't do everything for us. It's a tool.
Just one. It's like the chess players who, the chess Zero, that program that has surpassed all the greatest chess players. They see what it does, but they can't imitate it. So they can't fully understand what it's doing, but it's way ahead of what they can do.
Yeah, and the interpretation of AI system is a whole research field, because it does things, but we don't really know how it does things. And so then you need to sort of open the hood of the AI system and try to understand what it does by feeding it some special input and see how it reacts. And it's an interesting field. It's an interesting topic.
Thanks very much. Thank you.
So just to kind of sum up, if there's some projects where you, within the same individual, if you do it over and over again, you might be able to say, okay, this person from the data was looking at a human face versus something in nature. Like there's some evidence showing that you could read those patterns if you test the person over and over again. So conceivably, if that's translated to remote viewing, you might be able to look at the patterns and say the target is possibly a person as opposed to a tree, because the data looks like this when it's human and it looks like this when it's a tree. But if you are going to do that, you might need like thousands of trials within that individual. Is that pretty much like what you're saying? Like that might be a worthy study, but
Yeah, exactly. Yeah, exactly. I mean, probably I know people have done that with fMRI. So they ask the person, imagine a tree, imagine a person, or something different, right? And then they can decode that information. So I guess you could do that, remote viewing. But yeah, again, you'd need many trials. With EEG, it's a bit more tricky, I think, than with fMRI because the signal is more noisy. But yeah, I think it would be potentially possible. Mm, Potentially, yeah, but probably with difficulty as well, because the remote viewing patterns might be very fleeting. You don't know exactly when they happen, potentially. Um,
And
but
so,
and yeah.
since there is at least one person out there right now claiming that she can do that, you would say you would want to see her data, like you would want to see this like
How they get to that? Yeah, how do they get to that conclusion? Yeah, yeah, because that's not something you can just come up with, right? By just observing. Oh, I've done. It looks like every time I do this, it's probably going to be very subtle signals that you probably cannot see on the naked eye, and that's why people have done like need to do many trials and because the signals, the changes in the signals are very subtle.
And do you think a person can effectively just test themselves repeatedly? Like if you're going to run this experiment, would they need someone like doing it with them, or could they conceivably just do it themselves? And like how many trials or how much time do you think would be required to for you to be where you'd be convinced that the patterns they're seeing are really there?
Um, yeah, I guess if you have all the skills to do it, you could do it yourself, I suppose. Probably you need as I said, like the software development kit. So you need to have your own software that would do it, and then. So if I was to do it myself, say I'm a remote viewer, I'd have to. So I don't know, many trials, many remote viewing sessions. Say I want to know whether when I see something, it's animate versus inanimate. So every time I see something inanimate, I would mark it in time, and then every time I see something that's inanimate, I would I have a time marker, and then I would cut the EEG signal every marker that I put, and then I would feed that into say a classifier, or maybe I can calculate maybe the power, the relative power. Um, yeah, I mean that's what I would do. I don't know if it would be really useful, to be honest, because why not directly rely on your own impressions then, rather than the EEG signal? That would be my question. So in what way the EEG signal will be more reliable than your subjective impressions? Your subjective impressions are what they are. Your EEG signal will be very noisy and much more difficult to interpret in many ways, I think. A lot of people get very excited about neurotechnology, about EEG and all these methods. And when people dig a bit more into it, or often they're quite disappointed, but because it tells us that they expected that it would tell them a lot about what's going on. But actually, those are very crude systems in a way. So the neurons that I showed there, on my schematic, the tiny. It's imagine you have a satellite outside that's like in the a satellite in the space that's listening to us talking. It's kind of the same things. Like if everyone's shouting at the same time, right? It's what the satellite would measure or something like that. Shouting actually maybe for waves if you want, but what I mean is that we're measuring very crude signals with EEG. It's just like a bunch of electrodes, but we have millions of neurons, hundreds of millions of neurons, and the signals go really fast, and the EEG is fast, but still it's probably too slow to really understand what to really measure things that are. I mean, because the signals are so large in a way, like so what the EEG measures is big population activity. So this is good. So it means that the signal that we're actually measuring are meaningful, but it's very crude, right? Um, what I mean is that there are very strong limitations in EEG, like every method that we have. We don't want to overinterpret things. We need to be very careful how we interpret things.
I'd
Andy,
like to say, sorry.
Andy put his hand up and then
Oh, yeah, no problem. Oh,
Hi.
yes, sorry.
then Andrew and then Dave, and then I think that'll be our final question. So yeah, Andy.
Yeah, thank you. One, just so you would be, I'm glad to hear there's going to be studies on this with remote viewing. One thing I just thought would be good to consider, which probably you already have, is that many of us experience perceptions that are not visual in nature, right? There could be like auditory type things and other perceptions. So it'd be really interesting to see a study of like if you do, trying to remember a visual of something versus remembering a smell of something, right? And how that shows up in the data. It might be too much in the noise of things, but it would be really, because a lot of us will perceive things that are outside of just like a visual perception. There's a lot of other ways, data can be received. So it'd be really interesting to see that. And my only other last question real quick is I've seen people experimenting in the remote viewing community using other off-the-shelf devices that help induce a certain brain state. Instead of reading your brain state, they claim it induces a brain state. And I was just wondering if you're familiar with those devices and have any personal opinions on how that might, maybe aid somebody in doing sort of work like this. Yeah.
So about the modality mentioned that you said, yeah, I think that would be interesting to compare. And I think there's been some research on, imagery, so visual imagery versus auditory imagery, things like that. I'm not so sure about EEG. I know it's been done in fMRI.
Okay.
And certainly it would be interesting to do it with remote viewing, because the difference is that it's not the images come to you, or the sensations come to you. It's not that you actively generate them, right? So it's a difference that's quite interesting. For the system that induces state, I don't know which ones you're talking about, so I don't know what it is. So with
Yeah.
Muse, what people try to, like, I think one of the claims of the Muse is it's kind of a biofeedback thing. So you see your alpha, so you try to learn how you can modify your state by seeing your brain signals. So I think there is this idea of trying to get better at getting into states. So what about the other signals, or the other? mm, Um,
I believe it's like a neorhythm device, and they claim it induces a certain brain state, like alpha or something like that.
how does
And
it work? Do you know how it works?
I don't know. It's like a PEMF type. I'm not sure. If you're unfamiliar, I don't expect you to reply to be
No,
able to
sorry.
respond to it. But I just was curious if you had heard of such devices and if there was any. So yeah, no worries at all.
I
So
don't know.
yeah,
I don't know about that. I don't know.
yeah. Thank you though. Thanks.
And people have brought up HemiSync. Steve, did you want to share something as far as I saw something in the chat, but at Monroe, they've developed sound systems that are supposed to put your brain into the different states. And I know they're still in the process of really needing to conduct more formal research on that as well. So I think that's what people are referring to there. But the HemiSync isn't measuring your brain. It's supposedly bringing your brain into synchronization and states that are thought to be conducive to have these other experiences. Well, and just this makes me think of the study that we're hopefully going to have published soon on the IRVA survey on do remote viewers know when they're on target or not, or when they're correct about a piece of information. And we found that about 87% said on occasion they do have a strong feeling of when they're correct and it turns out to be correct. And so whether we're talking about using equipment or we're talking about just viewers, being able to tell within themselves, like I feel particularly relaxed when I'm on target, or I feel particularly excited, or I get this, my heart starts to beat fast, or, being able to tune into oneself and start to recognize the body signals or the patterns. I think that's what we're, really kind of talking about here, whether it's our own sensing system or we're being able to use some equipment outside ourselves. But I think that's kind of the ultimate goal, is so that we can achieve greater accuracy or understanding. Well, with that, is there anyone else who had any final pressing question, or just, I know we had some things in the chat we might have overlooked, but any final words
I would
before?
like to say one thing. I hope it's okay to say, but talking about using different tools to like when I was very active with APP, sometimes I would do the remote viewing, but then when they were giving before we self-judged and then they would put out the results. As an intuitive, I could read the results and know if those, not 100% of the time, but occasionally I could read the results and know if the result was a winning bid or not. So it was just another tool in the same way that we could use Muse or kind of anything. That's all I want to say.
Yeah, and that does get back to what David was saying, that it might just be more efficient to be relying on our intuitive impressions than trying to get this equipment to register something to confirm what we're already getting, or on that matter, having multiple viewers or multiple intuitives giving their input at the same time. But thank you so much, David, for all this time. I know it's later there for you and your weekend, and so very much appreciate that you came and talked to us. This was extremely informative, and we have recorded it so we can get it out there on our YouTube page. And just want to thank everyone for all of your questions and for being here. And it's so nice to see some of you. Nancy, haven't seen you in so long. Nancy was on IRVA's board for 5 years, and she's a good friend too. So it's great to have you here. And yeah, so we're a little bit over our time. David, thank you again.
Oh, you're welcome.
Any final words you'd like to say, or you've probably said enough?
No,
But
it's fine. I'm yeah, I hope that was informative, that you learned a couple of things and that was useful for you. And yeah, and that's it. And thanks for attending and for inviting me.
yeah, you're very welcome. And it'll be really fun to have you come back, whether here or at a IRVA conference or somewhere when you're ready to report on your research. You're going to have a lot of interest. So that'll be great to get updates. And yeah, okay, well, so next month we'll have our meeting the first Sunday of the month. Hopefully you guys will come back, and then yeah, I think we'll just sign out for now. Dale, if you can hang out for a minute, and yeah, Nancy, if you could hang out too, that would be great for a minute. And then we'll see everybody next time. And I'm going to stop the recording here.
All right. Goodbye.
Thank
Thank you.
you, everybody. Have a great month.
Thank you.
You too. Thanks for doing all.